Imagine just a microscopic speck of dust landing in your eye.
Right.
To you, it's like nothing but a minor annoyance. You blink, maybe rub your eyelid and just go about your day.
Yeah, totally.
But beneath the surface, on a cellular level, that tiny intrusion just triggered this highly coordinated, incredibly violent military response.
Oh, absolutely.
And when you look at the clinical immunology materials we brought in today, you aren't just reading some dry medical textbook. You are looking at the literal blueprints for that invisible war.
You really are.
And our mission today for this deep dive is to translate these wildly complex schematics, the actual flowcharts of the human immune system, into a vivid, really easy to understand experience for you. By the end of this deep dive, the next time you get like a simple paper cut or you feel that first familiar sniffle of allergy season, you are going to be able to picture exactly what is happening inside your tissues.
Down to the molecular level.
Right. So to start, before we can decode the dense flowcharts of how your cells fight, we kind of have to understand the geography of where they live.
The battlefield map.
Exactly. You've got the innate immune system and the adaptive immune system.
And that division is really the foundation of everything.
Right.
The innate side of the system is what you were born with. It's pre-programmed and incredibly fast. These schematics show this branch starting with sheer physical barriers.
So like the literal walls.
Right. We're talking about the tightly packed keratinized cells of your skin or the sticky mucous membranes lining your lungs and your gut.
Oh, wow.
But it also includes these fast-acting generic responder cells, things like neutrophils and natural killer cells.
Okay, got it.
Then on the complete opposite side of the map, you have the adaptive system. This is where your highly specialized T cells and B cells live.
And they're housed within this vast, sprawling lymphatic network, right? Like the nodes, the spleen, and the thymus.
Exactly.
Okay, let's unpack this setup, because looking at how these two sides operate, it feels exactly like the defense strategy for a massive medieval castle.
Oh, I like that.
Right. So the innate system, that is your moat, your thick stone walls, and the immediate heavy armor frontline guards standing right at the drawbridge.
Yeah, they are brute force.
Exactly. They react the exact same way to every single intruder, no matter if it's a splinter or like a deadly virus. They don't ask questions, they just attack.
Right.
But the adaptive system, well, that operates completely differently. That is the elite clandestine intelligence agency operating deep inside the innermost keep of the castle.
They're the spies.
Exactly. They might take days or even weeks to study an intruder's specific profile, but once they identify the threat, they custom design a targeted weapon specifically for that one enemy.
That analogy holds up perfectly, especially when you look at how the diagrams show these two branches communicating.
Oh, right, because they have to talk to each other?
Yeah, the intelligence agency deep in the castle needs to know what the frontline guards are actually fighting out there.
Right.
So you have this network of lymphatic vessels.
The highways.
Right. If you look at an anatomical map, they look like a second circulatory system running totally parallel to your blood veins. And these vessels act as a massive one-way highway. They're constantly sweeping up interstitial fluid, which is the fluid bathing your tissues, say, from that paper cut on your finger.
Wait, so it's essentially acting as a vacuum, like just sucking up the battlefield debris?
Precisely. It sweeps up that fluid and channels it away to the lymph nodes.
Which are the spy headquarters.
Exactly. The lymph nodes are the intelligence agency's headquarters. They're packed tight with those elite T and B cells, which are just, constantly sampling the fluid flowing in.
Inspecting the debris.
Inspecting the debris, yeah. Checking to see if the frontline guards at the paper cut have encountered any foreign invaders that require a specialized custom-built response.
So the front line is constantly sending intelligence back to headquarters. That's just a phenomenal system.
It really is.
But I want to zoom in on those frontline guards for a second, because the material outlines the exact flowcharts for how these cells neutralize a threat in real time, and it's fascinating.
Oh, the phagocytosis diagrams?
Yes. There's this sequence depicting phagocytosis, which is essentially the cellular version of a horror movie. It maps out how a white blood cell literally eats a live bacterium.
Yeah, phagocytosis is the core mechanism of the innate system. A cell, like a macrophage, which literally translates to big eater, by the way.
Big eater. That's great.
Right. It engulfs and chemically digests a microorganism. But the diagrams show us that this isn't just a blind Pac-Man-style eating process.
Right. It's not just wandering around eating everything.
No, the surface of the macrophage is bristling with these incredibly sensitive sensors. The text refers to them as pattern recognition receptors.
Okay, yeah, I noticed these acronyms in the charts like toll-like receptors and NOD-like receptors, but what are they actually sensing? Are they like eyes?
Think of them more like biological barcode scanners.
Oh, barcode scanners. Okay.
Yeah, they're constantly scanning the environment for molecular patterns that simply shouldn't exist in the human body.
So they don't know what it is exactly.
Right, they don't know exactly what the specific bacteria is, but the scanner reads a molecule like peptidoglycan, which is a major component of bacterial cell walls, and immediately flags it as non-human.
Wow. Okay, so the alarm goes off, but the flow chart shows these extra tiny little bridge structures wedged between the macrophage and the bacteria during the eating process.
Yeah.
The text labels them as opsonins. What is the biological logic there? I mean, if the cell already recognizes the bacteria, why does it need a bridge?
Well, because recognizing a threat and physically grasping it are two very different things.
Oh, interesting.
Yeah, bacteria have evolved to be incredibly slippery. Many are actually coated in this slick sugar capsule, specifically designed to evade capture.
Sneaky.
Right. Imagine trying to catch a greased pig with your bare hands.
That sounds highly inefficient.
Exactly. It's highly inefficient. So opsonins are the solution. They're specialized proteins like one called C3b, or even antibodies that just float around and attach themselves firmly to the surface of the slippery bacteria.
Oh, so they act like handles. The macrophage's receptors grab onto the opsonin handles instead of the slippery bacteria itself.
Exactly. The handles allow the cell to get a firm grip, making the whole engulfment process vastly more efficient.
That is so smart.
It is. And the mention of that C3b protein actually brings us directly to one of the most mechanically complex flowcharts in the entire system.
Oh, boy. The complement cascade.
The complement cascade.
Yes, let's dive into this because, wow. Looking at the schematic for the complement pathway, it looks like a literal cascade of falling dominoes.
It's intense.
It is so intense. You had these three completely different starting arrows at the very top of the chart. There's the classical pathway, the lectin pathway, and the alternate pathway.
Right.
And they all tumble down, triggering these different chemical reactions until they all just crash together into one single central node labeled C3.
And C3 is the critical junction.
Okay.
When that C3 domino falls, it splits. One part becomes that handle we just talked about to help macrophages eat the bacteria.
The C3b handle.
Exactly. But the rest of the flowchart plummets straight down into the assembly of something called the MAC, or the membrane attack complex.
The membrane attack complex sounds aggressive.
It is very aggressive. This is a sequence where proteins, numbered C5 through C9, physically assemble themselves into a microscopic cylinder and literally punch a massive hole directly through the bacterial cell wall.
Wait, it just drills a hole right into the enemy.
Yes, it just punches right through the armor. And the mechanism of death here is osmosis.
Oh, because of the pressure difference.
Right, because there is now a gaping hole in its armor. Fluid from the surrounding environment rushes into the bacteria. It loses all osmotic pressure control, swells up, and it essentially explodes.
Wow, it just pops them. It's such a brutal mechanism.
Highly effective, though.
True, but I have to admit, like, looking at the layout of this complement cascade, I find myself really questioning the design.
How so?
It seems wildly, almost dangerously complicated. Like, why have three completely different starting pathways at the top of the chain if they all just funnel into the exact same C3 domino anyway? Why not just have one simple tripwire to start the whole process? It seems like a lot of wasted energy.
Well, you're touching on a fundamental principle of biological survival here, which is evolutionary fail-safes.
Okay.
The immune system is locked in a never-ending arms race with pathogens. If you only have one tripwire and a pathogen evolves a way to step over it, you die.
Right. Game over.
Exactly.
Right.
For instance, the classical pathway at the top of that chart requires an antibody to bind to the bacteria to start the dominoes.
Okay.
But what if it's a brand new mutated bacteria and your body doesn't have an antibody for it yet?
Oh, then the classical pathway is totally useless. The dominoes don't fall.
Right, and that is exactly why the body evolved the lectin pathway. It operates as a backup alarm that triggers by recognizing specific foreign sugars on the microbe's surface, totally bypassing the need for an antibody. And if the bacteria somehow hides its sugars, well, there is the alternate pathway, which triggers directly upon physical contact with the bacterial cell wall itself.
That is brilliant.
It's a flawless example of protective redundancy. If the bacteria evades one tripwire, there are two more waiting in the dark to ensure those dominoes still fall and the threat is neutralized.
So redundancy is the ultimate lifesaver on the front line.
Absolutely.
That makes total sense. But that brings up a glaring issue regarding that elite intelligence agency we talked about earlier, the T cells and B cells.
Okay.
If the innate system is all about triggering as fast and as often as possible, how does the adaptive system deploy its massive custom-built weapons without accidentally declaring war on our own body?
That is a million-dollar question.
Because the flowcharts detailing adaptive cell activation are easily the most convoluted restrictive diagrams in all of the source material.
And they have to be restricted. I mean, the adaptive system has the power to cause systemic, irreversible destruction.
Right.
If a T cell or B cell misidentifies one of your own healthy heart or joint cells as a foreign target, you develop a devastating autoimmune disease.
Which is terrible.
Right. So the rules for their deployment have to be absolutely ironclad.
Let's break down the logic of those rules then, starting with the T-cell activation sequence.
Sure.
The diagram shows a very specific two-part requirement for a T-cell to wake up. It needs what the text calls signal 1. Now, as I understand it, signal 1 happens when the T-cell's receptor locks onto a shredded piece of the enemy, an antigen.
Correct.
But the diagram shows this piece of the enemy is being presented to the T-cell by another cell using something called an HLA molecule. Can we translate that?
Yeah, think of the HLA molecule as a molecular flagpole.
Flagpole.
The presenting cell, say, one of those frontline macrophages that just ate a bacteria, chops up the invader, takes a tiny recognizable piece of it, and hoists it up on this HLA flagpole on its own surface.
Look what I found.
Exactly. Yeah. It then travels to the lymph node to show the T cells. The T cell inspects the flagpole. If its receptor matches the piece of the invader, boom, that is signal 1.
But the flowchart shows a massive stop sign right there. Like, the activation halts. The T-cell refuses to arm itself unless signal 2 happens at the exact same millisecond.
Exactly. Signal 2 is a co-stimulatory signal. It's essentially a biochemical handshake.
A handshake.
Right. The presenting cell doesn't just show the flagpole. It also extends a separate molecule, usually one called CD80 or CD86.
Yeah, okay.
And the T cell has to grab that molecule with its own receptor called CD28. If the T cell sees the enemy on the flagpole but does not get that second handshake, it doesn't just stay asleep.
What does it do?
It actually assumes something is terribly wrong and forces itself into a state of permanent paralysis.
Permanent.
Yeah, a condition called anergy.
Wow. This logic is fascinating. I mean, it's identical to the security protocol for a real-world nuclear submarine launch sequence.
Oh, that's a great way to look at it.
Yeah. The military demands that 2 separate commanding officers stand across the room and turn 2 completely different keys at the exact same time to launch a missile.
Right.
If you only have one key, or, one officer goes rogue and tries to fire, the system doesn't just pause, it totally locks down to prevent a catastrophe.
That is precisely the mechanism. It is a biological 2-key security protocol.
Man.
And what's really striking is that the flowchart for B-cell activation utilizes a nearly identical safeguard.
Really?
Yeah. A B-cell can sometimes recognize an enemy antigen on its own, but to fully activate, to undergo what's called clonal expansion.
Where it rapidly clones itself into a massive army of cells pumping out millions of antibodies.
Exactly.
Yeah.
To do that, it almost always needs a T helper cell to come over and insert a second key.
Wait, so the T cell has to verify the B cell's target?
Yes. The diagram shows the T cell using a molecule called the CD40 ligand to bind to a CD40 receptor on the B cell.
Okay.
That connection is the second key. Without it, the B cell cannot launch its antibody strike.
2 keys to launch the missiles. Always 2 keys. Always. But surely, even with 2 keys, things can get out of hand once the war starts, right? I mean, how does it stop?
Oh, they absolutely can get out of hand, which is why the blueprints also contain built-in emergency brakes.
Brakes. Okay, I saw these.
Yeah, as you look through the activation pathways, you'll see inhibitory molecules depicted. They're receptors with names like CTLA4 and PD1.
Right, I saw this in the charts, but how do they work mechanically? Do they just unplug the keys?
Kind of. They act as competitive inhibitors. Remember that second key handshake we talked about with the CD28 receptor?
Yeah.
When a T cell has been fighting for a while and it's time to cool down the immune response, the cell starts producing CTLA4.
Okay.
And CTLA4 actually binds to the exact same spot on the presenting cell, but it holds on much tighter.
Oh.
Yeah, it essentially steals the keyhole, it blocks the activating handshake, and forces the T cell to stand down.
It just shoves the other key out of the way.
Exactly. Without these incredibly complex restrictive breaks, the immune system would just keep firing until it caused massive collateral damage to your healthy tissues.
Okay, so we've seen these visual flow charts working perfectly. We've mapped the castle architecture, we've watched the backup dominoes fall to punch lethal osmotic holes in bacteria,
Right.
and we've decoded the nuclear level security required to launch an antibody strike. But let's pivot to the dark side of this system.
Okay, let's go there.
What happens when this precise chain of command, this incredibly heavily armed military, is triggered by something utterly, completely harmless? Like a tiny protein in a peanut or a speck of pollen.
Ah, that takes us into the clinical realm of hypersensitivity, specifically type I hypersensitivity.
Okay.
The diagrams mapping out an allergic reaction show a profound, cascading misfire of the exact flowcharts we just discussed.
Let's walk through the mechanics of this one because the cause and effect mapping here is wild.
It really is.
The flowchart shows a timeline starting with a first encounter with an allergen. So let's just use the peanut example.
Sure.
The chart shows the B cells and T cells activating. The keys turn, but instead of producing standard defense antibodies, they produce a very specialized class called IgE.
Right.
And instead of attacking the peanut, these little Y-shaped IgE molecules just float away and embed themselves firmly into the surface of a completely different cell called a mast cell.
Yes. And that positioning is everything. By embedding themselves onto the exterior, they basically turn the mast cell into a submerged sea mine.
A sea mine.
Completely coated in biological tripwires, just waiting. The mast cell is now officially primed.
Okay.
And the diagram for the re-encounter with the allergen, that is where the systemic catastrophe happens.
Right. The second time the person eats a peanut, the proteins enter the body and hit those IgE tripwires. Now, previously, I might have just pictured the cell exploding like a piñata.
Sure.
But the text explicitly focuses on a mechanism called cross-linking. What is actually happening there?
It's all about physical tension. Yeah. A single peanut protein molecule is relatively large enough that it actually binds to two separate IgE antibodies on the mast cell at the exact same time.
Oh.
So it acts like a bridge between them, physically pulling them together. That pulling action is the cross-linking.
So it's literally yanking two tripwires simultaneously.
Exactly. That physical yank is what pulls the pin on the mast cell. The cell membrane forcefully degranulates, bursting open and dumping a massive payload of inflammatory chemicals directly into the surrounding tissue.
And the most prominent chemical in that payload is histamine,
You guessed it.
which connects us to the clinical diagram of anaphylaxis, because the flowchart tracks the arrows from that single burst of histamine straight to immediate, system-wide, life-threatening effects on the human body.
The clinical effects are devastating, mostly because of how histamine interacts with different tissues.
Right.
The first arrow points to extreme vasodilation.
Yeah.
Histamine forcibly relaxes the smooth muscle around your blood vessels, causing them to open up really wide.
But why would the body want to do that in the first place?
Evolutionarily, if you have a parasite in your tissue, opening the blood vessels allows a massive rush of white blood cells to flood the area and fight it.
Oh, that makes sense.
But during anaphylaxis, this happens system-wide. When all the blood vessels in your body suddenly widen at once, your blood pressure completely crashes.
The bottoms out.
Yeah, the clinical chart labels this as severe hypotension or circulatory collapse.
So there literally isn't enough pressure to pump blood to the brain.
Exactly.
And then the diagram shows another arrow pointing to the lungs, showing bronchoconstriction.
Right. Histamine causes the airways in the lungs to violently tighten and spasm.
Yikes.
Again, the ancient evolutionary logic was likely to prevent breathing in more of a toxic substance or parasite, but the clinical reality is a severe asthma-like attack, basically cutting off oxygen.
And the third arrow shows fluid leaking out of the vessels and into the skin layers, which causes urticaria.
Or hives, yeah.
Right. Hives, again, the mechanics of this pathway exposure, IgE production, cross-linking the tripwires, and then the histamine dump causing systemic collapse. So it's all perfectly logical when you break down the biology.
It is.
But here's the major question I have for you. Since the mechanics of this pathway are so perfectly straightforward, why doesn't everyone's immune system flowchart map a peanut as a deadly threat? Like, what dictates who gets this misfire and who can just eat a peanut butter sandwich every day without an issue?
It's honestly one of the most compelling questions in modern immunology. And if we look at the source text, it explores this mystery by moving beyond genetics and looking at our environment.
Okay.
The text highlights a concept known as the hygiene hypothesis.
Okay, let's pause and unpack that. What does hygiene have to do with immune flowcharts?
Basically everything.
Really?
Yeah, the hypothesis suggests that human immune systems evolved over millions of years anticipating a heavy, constant load of childhood infections.
Like,
We're talking about parasites, worms, and constant bacterial exposures.
mm,
Our immune flowcharts, especially those Ig pathways, were designed to be incredibly busy fighting off these large invaders.
oh, I see where this is going.
Right. But in modern developed environments, we have sanitized water, widespread antibiotics, and indoor living. The burden of early childhood infections has been drastically reduced.
Wait, so because of modern hygiene, the immune system is essentially just sitting around bored.
In a mechanical sense, yeah. Without that early, heavy combat training against real dangerous pathogens, the immune system's regulatory networks, those complex inhibitory brakes we talked about earlier, they don't develop the proper bias.
The flowchart gets miswired.
Exactly. It gets miswired during childhood. Instead of learning to ignore harmless environmental proteins like pollen, pet dander, or food, the untrained immune system incorrectly labels them as high-level parasitic threats.
Wow.
It defaults to that ancient, aggressive IgE mast cell pathway because the filthy environment it was built to operate in has suddenly radically changed.
That absolutely blows my mind. The idea that our sterile, safe, modern environment is actually rewriting the sensitivity of our internal defense blueprint.
That's wild.
It's misdirecting millions of years of evolution.
It's a powerful reminder that these flowcharts we are studying aren't static, rigid documents.
Right.
They are highly reactive, deeply adaptable systems that are constantly in conversation with the world around us.
So to bring all of this together for you listening, we started today by looking at the macro map, right? The geography of the immune anatomy.
The innate moats and walls.
Exactly, and the lymphatic highways constantly sweeping intelligence back to headquarters. We zoomed in to watch the innate domino cascade of the complement system, learning how evolutionary redundancy ensures we can punch lethal osmotic holes in bacteria.
Poor bacteria.
Right. Then we deciphered the nuclear launch, two key security handshakes of our T and D cells, which are designed with complex brakes to keep us safe from friendly fire.
Essential breaks.
And finally, we tracked the tragic systemic misfire of anaphylaxis, where a bored immune system cross-links a tripwire and turns its devastating ancient arsenal against a simple peanut.
We've effectively translated those dense 2D schematics into the dynamic, violently beautiful living system that is keeping you alive right now.
We did. But before we wrap up, there is one final incredible detail buried in the clinical text that we need to touch on.
Oh, the biologic therapies.
Yes. The source notes that modern biologic therapies, these cutting-edge immune drugs, are actually being used to target singular, specific nodes in these flowcharts.
It's true.
For example, doctors can now prescribe drugs that specifically neutralize TNF-alpha to stop runaway rheumatoid arthritis, or they prescribe drugs that intentionally block that CTLA-4 brake we talked about.
Effectively cutting the brakes to unleash the immune system's full fury against cancer cells.
Which represents an unbelievable leap forward in medicine.
For the first time in human history, we are no longer just treating the outward symptoms of a disease. We are manipulating the foundational diagram itself.
Right. We are acting as editors. We are literally erasing or drawing new chemical arrows in our body's ancient immune blueprints to cure diseases.
It's incredibly powerful.
But understanding how interconnected all these dominoes are leaves me with this thought for you to mull over.
Let's hear it.
If human medicine can now artificially force these millions of years old dominoes to fall, or surgically prevent them from falling, what new, entirely unforeseen chain reactions might we be accidentally writing into our own biology in the process?
A profound question. I mean, when you alter the structural pillars of a blueprint that took millions of years to draft, you have to be prepared for the entire building to settle in highly unexpected ways.
Something to think about the next time you get a paper cut.